Hello guys, so today we are going to discuss about pteridophytes. Before starting the subject, hi, how are you all? I hope everyone are doing great. Myself Ujjwala and welcome back to the channel with me Ujjwala. I hope everyone are doing really great and yeah, if you have not yet subscribed, please like, share, subscribe and become a part of the family. And yeah, without any delay, let's get into the video. So today we are going to discuss the topic pteridophytes. So the pteridophytes includes horsetails and ferns. So ఈ టెరిడోఫైట్స్లో మనం మెయిన్గా ఇంక్లూడ్ చేసేది హార్స్ టైల్స్ అండ్ ఫర్న్స్ అనమాట. దీస్ ఆర్ ఇంక్లూడెడ్ ఇన్ ద టెరిడోఫైట్స్. టెరిడోఫైట్స్ ఆర్ these pteridophytes are of medicinal purpose and as soil binders. They are frequently used as ornamentals. So pteridophytes యొక్క యూజెస్ ఏమంటే అవి మెడిసిన్లో యూజ్ చేశారు, ఇంకా దే విల్ బి ఆల్సో యూజింగ్ దెమ్ యాజ్ సాయిల్ బైండర్స్ అనమాట. అంటే ఎరోషన్ ప్రివెంట్ చేసి సాయిల్ అనేది ఎరోషన్కి గురి కాకుండా బైండ్ అయ్యి ఒక చోట ఉండడానికి, అప్పర్ సాయిల్ ఎరోషన్ అనేది జరుగుతుంది కదా, సో టాప్ సాయిల్ ఎరోషన్ జరగకుండా సాయిల్ బైండ్ చేసి పెడతాయి అనమాట. టు ప్రివెంట్ ద ఎరోషన్ దీస్ pteridophytes ఆర్ యూజ్డ్ అనమాట. అండ్ ఆర్నమెంటల్స్ టు ఎన్హాన్స్ ద బ్యూటీ ఆఫ్ సంథింగ్ లైక్ ఒక రూమ్ కార్నర్కి బ్యూటీ ఇవ్వాలన్నా, రూమ్ ఎలివేటెడ్గా లుక్ చేయాలన్నా, దీస్ pteridophytes are used as ornamental plants like money plant, etc. and etc. It's an example, okay? So these are used as ornamental, so in enhance చేయడానికి బ్యూటీని. And evolutionarily they are the first pteridophyte plants to possess vascular tissue. Vascular tissue అనేది ఫస్ట్ ఇట్ ఈస్ సీన్ ఇన్ ద pteridophytes అనమాట. ఇంతకుముందు బ్రయోఫైట్స్లో లేదు, ఆల్గేలో లేదు. ఓన్లీ ఫ్రమ్ ద pteridophytes we will be learning about the vascular system. So first terrestrial plants to possess vascular tissue. Vascular tissue అంటే xylem and phloem. What is xylem? The tissue which carries the water is known as xylem, and phloem food and mineral carrying tissue is known as phloem. So దీంట్లోనే మనం ఫస్ట్ vascular tissuesని చూసింటాము pteridophytesలో. You shall study more about this in the chapter 6. The pteridophytes are found in the cool, damp, shady areas. Where are they found? Cool, damp, shady areas. Some may also flourish well in the sandy soil conditions also. So cool, damp, shady areas lo majority of the pteridophytes are found, and some may even flourish well in the sandy soil as well. So these pteridophytes are examples are hostels and ferns, already discussed. These are used for medicinal purpose. The soil binders, ornamentals, okay, well these are the first terrestrial plants to possess the vascular tissue like xylem and phloem, okay, very good. Where are they found? They are found in the cool, damp, shady areas, and also they flourish well in the sandy soil conditions as well. Fine. Now you may recall that in bryophytes the dominant phase is the gametophyte. So till now what we have discussed in the all the previous algae, bryophytes, pteridophytes, gymnosperms, angiosperms, right? In algae, bryophytes, we have seen that the dominant phase is gametophyte, which is a haploidtic in condition. While it comes to pteridophytes, in the pteridophytes the dominant one is, in bryophytes the dominant one is gametophyte, right? But in pteridophytes it is sporophyte. Okay, in the pteridophytes it is sporophyte, though the life cycle is haplodiplontic. This is very important. Pteridophytes lo manaku I want to show you it in detail because it is very important. So let me take a piece of paper and discuss this. Okay, now don't mind. Okay, now when it comes to algae, bryophytes, pteridophytes, gymnosperms, angiosperms, right? In the algae we will see haplontic life cycle, N condition, and main plant body is gametophyte, right? Gametophyte. But when it comes to bryophyte, it is all in bryophyte we will see haplodiplontic, but the dominant one is also N, so main plant body is gametophyte. Okay, now when it comes to pteridophyte, though it is haplodiplontic, it shows 2N in the sense the dominant one is sporophyte. So dominant stage is sporophyte. That is what I want to discuss to here. In gymnosperms the life cycle is diploid, 2N condition, sporophyte. So when it comes to angiosperms also it is diploid, 2N condition, it is sporophyte. Okay, I hope this is understood by you. Okay, now here that is what they have given. You may recall that in bryophytes the dominant phase is cycle is gametophyte. In the bryophytes, which I have told you, bryophytes, gametophyte is dominant, it is in condition haplodiplontic, though haplodiplontic, dominant is gametophyte, right? But in the pteridophytes main plant body is sporophyte, right? In the pteridophytes main plant body is sporophyte, though the haplodiplontic, main plant body is sporophyte. Okay, now sporophyte, which is differentiated into, so here the sporophyte is differentiated into true stem, leaf, and roots. So which we have seen in algae, the body is thalloid body. In bryophytes, first embryo formation is seen. In pteridophytes, vascular tissue is seen. In gymnosperms, we will be seeing seeds formation, and in the angiosperms we will be seeing the fruit formation, right? Here the body is not thalloid, it is well differentiated into roots, stems and leaves. But in bryophytes, root like, stem like and leaf like bodies are present. When in algae, thalloid, total thalloid, okay? But here we will be seeing a well differentiated root like, stem like and leaf like plant body is seen. The organs possess well differentiated vascular tissues. So xylem, phloem are well differentiated in pteridophytes. The leaves in pteridophytes are small, microphyll. Micro in the sense small, right? Microphyll is very small in size. They are either small in size or large in size. Example for small microphyll, these leaves, example is Selaginella. Selaginella is possessing very small leaves, whereas ferns are possessing very large leaves. Okay, now microphylls in the sense leaves. Okay, now the sporophyte, the sporophytes bear. These sporophytes bear sporangia that are subtended leaf-like appendages called as sporophylls. So these are having leaf-like appendages. Those are known as sporophylls. When we see it in the gymnosperms, cones formation is seen. But here the sporophytes bear sporangia in leaf-like appendages. Okay, you have to differentiate that. When it comes to, let me show you. See, when it comes to this is pteridophytes, right? In pteridophytes, here the what I'm going to say is the sporophytes be or arranged in the sporangia that are subtended by leaf-like appendages. This sporangia is arranged in the form of a leaf, okay? But in the here leaf-like it is present. But in the gymnosperms, it is arranged in cones, okay? This is the main difference I want to tell you. This is the main difference, okay? Now, but here a few exceptions are there. All are not in the shape of leaf, called sporophylls. All sporophylls are not in the shape of leaf appendages. In some cases, sporophylls may form distinct compact structures. When all the leaves come together, they form a compact structure. It is known as cone. This cone is seen in only few in pteridophytes, but in gymnosperms, all forms cones. Here in pteridophytes, without a leaf, without cone formation, few are in the just like a leaf appendages. What these sporophylls? So those which forms distinct cone-like structures, for examples of those are given in NCERT. In some cases, sporophylls may form distinct compact structures called as strobilli cones. Strobilli cones, example for this distinct structure cone formation seen are in Selaginella equisitum. Very, very important. In pteridophytes, normally leaf-like appendages of sporophyll are seen, but few specialized cases are seen in the Selaginella and equisitum, which is very important. Now, this sporangia produces spores by meiosis in the microspore mother cell. Now, this sporangia, whatever is there, this sporangia provides spores. Obviously, it provides spores through meiosis, reduction division. It provides spores in the microspore mother cell. The spores germinate, gives rise to. Now, you have to get again, you have to get one point clear about this topic, which is, now, this is sporophyte. Right, this sporophyte, it is 2n condition, it possesses sporophyll, right, which is also 2n condition. It produces sporangia is present, right, it is also in 2n condition, right, sporangia is also in 2n condition. In sporangia, we will be having spore mother cells, okay, right. In the spore mother cells, we will be having reduction division, which is also known as meiosis. Right, through meiosis, spores are produced, right, spores are produced. These spores germinate to give what? They germinate to give gametophyte. That gametophyte is known as prothallus, okay, that gametophyte is known as prothallus gametophyte. This gametophyte possesses both the male and female gametes, and if this male and female gametes fuse together, again they form male gamete is known as antheridium, and the female gamete is known as archegonia. Okay, this is male gamete, this is female gamete. When these fuse together, they form zygote. This zygote develops into embryo, and this embryo will in turn gives rise to sporophyte. develops into sporophyte, and this process or this cycle continues. Here you can see the dominance of sporophyte, right, than gametophyte. So gametophyte period is reduced and sporophyte period is more. So that is why there is a dominance of sporophyte in the period in the pteridophytes. I hope this is clear. Okay, fine. This is the life cycle of pteridophyte. Okay. Let's move on. Now here the spores germinate to give rise to inconspicuous small multicellular next page, free living. This is small, inconspicuous, free living and photosynthetic thalloid. Now what is meant by thalloid? Here the gametophyte is in thalloid condition, in the sense it is not having true roots, stem, leaves. The body is undifferentiated, and gametophyte called as prothallus, which we have seen here. This gametophyte is known as prothallus. Now this prothallus will grow in cool, damp, shady areas, and because of the specific, this mainly grows in cool, damp, shady areas. So it does not grow everywhere. Only when these conditions are justified, this grows well. So because of the specific restricted requirement, the need of water for fertilization, very important. In order this antheridia go and reach to the mouth of archegonia, these are basically present in water. So this anthrozoids which are released, this should reach the archegonia, and in order to reach that, they have to swim in water, right? So water is very important for sexual reproduction in pteridophytes. So that is why, and water for fertilization, the spread of living pteridophytes is limited. So these are not present everywhere. They are only present in very few areas and restricted to narrow geographical regions. Narrow in the sense in very less geographical regions. The gametophyte bears male and female sex organs called anthrozoid and archegonia, which we already know. The same gametophyte, it contains both antheridia and archegonia, both are present, and respectively. Water is required to transfer anthrozoids, the male gametes released from the antheridia, to the mouth of archegonia. Fusion of male gametes with the eggs present in the archegonia results in the formation of zygote, which we know. This when male gametes, anthrozoids, when it reaches the mouth of archegonia, they go fuse with the egg and the formation of zygote takes place, and zygote therefore produces multicellular undifferentiated sporophyte. So again after fusion, a multicellular undifferentiated sporophyte is going to be formed, and this sporophyte is the dominant stage, which you can see in the life cycle. Majority of this is occupied by sporophyte, right? That is why it is the dominant stage of the pteridophytes. Which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are similar. Similar in the sense what? Homospores. Such plants are called homospores. In general, Selaginella and Salvinia, which produces two kinds of spores, macro, large, micro, small spores, which are known as heterospores. Normally, majority of the pteridophytes, they form homospores, but few they form heterospores, which are Selaginella and Salvinia, which produce heterospores. The megaspore and microspore germinate to give rise to female and male gametophyte respectively. The female gametophyte in these plants retains the parent. So here the female gametophyte which is there, right, that is not going to move anywhere. It is present in as it is in the gametophyte itself, but the male anthrozoids, they are released out of the gametophyte and they reach to the mouth of archegonia. That is why when you see the structure of prothallus, it is heart-like structure. Upper region, these anthrozoids are present, this archegonia are present, and then the lower region, this anthrozoids are. These are archegonia and these are anthrozoid. These are hair-like rhizoids. So these are released into the water current, and through the water current they will be reaching the mouth of archegonia. Okay, that is how these are going to happen. Now these megaspore and microspore germinate to give rise to female and male gamete respectively. The male gametophyte in the plants return to the parental sporophyte for the variable periods. Even though after what I want to say is, after fertilization also, after completion of zygote formation, embryo formation, after few periods, this female gametophyte is going to be dead. Okay, but it will be sustained for variable periods. Okay, the development of zygote into the embryo takes place within the female gametophyte itself. Okay, this event is a precursor to the seed habit. It is very important and considered as an important step of the evolution. It is very important. It means this sentence is giving us that first seed was seen in pteridophytes. I'm sorry, this seed seed habit in the sense, this is the very important step for the evolution. Okay, without this we can't see seed formation in the gymnosperms. The evolution, right? These are algae, bryophytes, these are classes which we were studying. This is a period of evolution, that is what they have given here. This event is a precursor of the seed habit, considered to be as important step of evolution. Okay, fine. Now when we see about the classes of these pteridophytes, these pteridophytes these pteridophytes are divided into 4 groups. Those are, first group is for the divided into 4 groups. Celopsida. Next group is Lycopsida. Next group is Penopsida. And the next thing is Pteropsida. So these are the 4 groups of the pteridophytes, right? Fine. Now the examples to remember this, it's very easy. Obviously you feel very difficult, but this is the 4 classifications which are seen in the pteridophytes. And for the Celopsida example is Celatum. Okay, this is the example. Lycopsida example is Selaginella and Lycopsidium. Okay, now for the Penopsida, Equisetum is the example. For Pteropsida, Dioteris, Teris and Adiatum. Okay. So now, ఇప్పుడు మనము pteridophytes examples memorizing tips చూద్దాము. ఫస్ట్ మనము pteridophytes యొక్క క్లాసెస్ ఉన్నాయి కదా ఇక్కడ. ఈ క్లాసెస్ని మెమరైజ్ చేసేకి ప్లీజ్ పడో. పడో అంటే చదువు. ఓకే, ప్లీజ్ చదువు. ప్లీజ్ పడో. PLSP. ఓకే, స్టార్టింగ్ లెటర్స్ని కన్సిడర్ చేయండి. ప్లీజ్. Psilopsida. P అంటే Psilopsida. L అంటే Lycopsida. S అంటే Spenopsida. అండ్ P అంటే Pteropsida. రైట్? ప్లీజ్ పడవు. క్లాసెస్ మెమరైజింగ్కి ఇది ఎగ్జాంపుల్. ఓకే, P అంటే Psilopsida, L అంటే Lycopsida, S అంటే Spenopsida అండ్ పడో P అంటే Pteropsida. Psilopsida, Lycopsida, Spenopsida, Pteropsida. ఫైన్. క్లాసెస్కి మెమరైజింగ్ టిప్ ఇది. నవ్ వెన్ ఇట్ కమ్స్ టు టోటల్ ఎగ్జాంపుల్స్ ఆఫ్ pteridophytes, ఎన్ని ఎగ్జాంపుల్స్ ఉన్నాయో అన్ని క్లాసెస్ని పక్కన పెట్టి ద మెమరీ టిప్ ఇస్ పెన్ స్టాండ్ లో ఎరైజర్ అండ్ పెన్తో DNA స్ట్రక్చర్ గి. ఓకే, పెన్ స్టాండ్లో ఎరైజర్ పెన్తో DNA కన్సిడర్ చేయాలి. ఏ స్ట్రక్చర్ గి. రైట్? కొన్ని వదిలేయాలి, కొన్నే మనం తీసుకోవాలి. పెన్ స్టాండ్లో పెన్ Pతో సైలాటం. ఓకే, పెన్ సైలాటం. ఎస్తో సెలాజినెల్లా. అండ్ T పెన్ స్టాండ్లో ఎల్తో లైకోపిడియం. ఎరైజర్ ఈతో we will be writing Equisetum. అండ్ పెన్ Pతో టెరిస్తో. DNA ఏతో we will be writing. అండ్ ఇక్కడ మనము పెన్ తో రైట్ DNA రైట్. సి హియర్ Dతో Nతో Dతో we will be writing Diopteris. Sorry, I have not told you that one. And for Aతో we will be writing Adiatum. Okay, now pen stand low eraser and pen తో DNA structure గి. రైట్? పెన్ Xylotum, stand Selaginella, low Lycopsida, eraser E Equisetum, pen P Teris, D to Dioteris, A to Adiatum. Right? This is the memorizing tip which is useful for you, and I hope this video is beneficial. And if you really like, share, subscribe, and stay tuned to next video. Bye.